Americans are lucky. We have access to some of the best pharmaceuticals in the world. And the reason for this is because we have so many resources in this country devoted to drug development. Forbes estimates that pharmaceutical industries spend north of $80 billion a year on drug development. That's with a B. So the way that works is the first thing they do before a drug ever sees clinical trials, the first thing that happens is that drug gets put into cells, onto a flat layer of cells. And they use that to model how the drug is going to do in people. There's a problem with that though. If you've ever looked at a person, you'll notice quickly that they don't look anything like a flat layer of cells. And that's evident in the tests. There's a lot of drugs that work great in the tests and they don't work very well in people. And vice versa. It's even worse, really. There's drugs that don't work very well in the tests and so they get scrapped and they're not continued down the development pathway, but they actually could work in people. So that's a huge problem for pharmaceutical companies and for you. The pharmaceutical companies waste a lot of resources looking at the wrong drugs, while they also miss out on drugs that could really help you. So what they need is a test, not in humans, just a test with cells, that's more accurately able to see how well the drug is going to work in humans. The Forbes lab at UMass Amherst that I work in thinks we have a way to do that with microfluidics. Microfluidics is a compound word, micro, which means tiny, and fluidics, which means fluids. So essentially what it is we use a device that has really, really tiny channels that we flow liquid through. And we use this to model arteries in tissues. So you have fluid flowing past a little cluster of cells, and because of the cells being in a cluster like that, some of them aren't touching the fluid. And so when you flow nutrients and drugs past them, the drugs actually have to move through the cells to get to the cells in the back. And that's exactly how your body works. Not every cell in your body is connected to a blood vessel. They all have to coordinate with each other. And so introducing that element, we think, into our tests will enable the drugs to more accurately show us how well they're going to work in people. So what I'm doing is seeing if that's the case, if this device is actually able to model the way a drug will work in a human. So I have a panel of 7 special chemotherapeutic drugs that have major differences in how they work in the flat cell test that they use right now and in clinical trials. So what I'm doing is taking those drugs and testing them in this device and seeing if they are able, if the device is more accurately able to model how the drug will work in humans than the monolayer test was. So the eventual goal is hopefully to make this device and be able to give it to the people who are doing research, either pharmaceutical companies or to people funded by the government working on drug development. And this would enable them to make drugs faster because they spend less time working on drugs that aren't going to work, and it will potentially decrease the cost of making drugs because they waste less resources. So you can get your drugs faster and cheaper.